Let's get started. We have a very short business meeting. Welcome everyone. We're happy to have you here tonight. I think you're really going to enjoy our program. And I got to see a preview and it was very, very interesting. Tonight's plant of the month is mealy blue sage, Salvia farinaceae. And this plant is of special value to native bees. It's a great nectar source. It's a wonderful plant. It's good for hummingbirds as well. Very deer resistant. It's in the mint family and blooms all summer long. This is a very hardy plant. You can plant a single plant, but it really looks good in mass and it likes to spread. And I have heard that the true native mealy blue sage is better at spreading if you're wanting to cover an area. There's a couple of cultivars. One of them is white called Augusta Duelberg. And then there's a deeper blue one called Henry Duelberg. And neither of those spread as much. So anyway, this is a very hardy native plant. And anyone with a pollinator garden or is doing butterflies or hummingbirds or whatever, this is a great plant. Activities since our last meeting. Charles has been busy removing invasives. He's looking for a couple of volunteers. Now that the weather's getting cooler again, he's going to be back out there. He is continuing at Berry Springs Park. He's going to begin at Gary Park. He's got the trees mapped out. I don't know what else he's going to be doing there, but he's also working on 2 Texas land conservation preserves that are in Georgetown. And he has completed his initial invasive plant maps for these 2. And he said, let's see, he's already removed 70% of the invasive trees at Berry Creek Preserve. So give Charles Newsom, if you don't know how to contact him directly, you can get on our website, the Williamson County website, and there is a form. If you scroll down, if you get to the home page, scroll to the bottom, and there's a form. You can contact anyone through that form. In addition, we have been busy. Beth Irwin has been busy, is also going to need a couple of volunteers to help at Berry Springs Park and Preserve. She told me what she was going to do, but I forgot. I think it's just sort of some maintenance around the plants and at Berry Springs Park. She's also been watching out for the Nest and Spirit Rains, 2 of our other pollinator gardens that she's designed, installed, and is taking care of. There's ongoing work at the Georgetown Public Library and the pollinator garden at the Georgetown landfill, which is Marilyn Purrs and also Agnes Platino. And then unfortunately, we have canceled our level 1 class. Our apologies to everyone that wanted to take an LCP level 1. We thought we would be ready, but it just got pretty apparent that we weren't going to be ready for that class in time. And we didn't want to do a bad job. We would rather do no job and offer you the chance to move to another class. And we'll get back to you when we have another class. We're going to do a plant sale at the farmers markets again this fall. Wildflower Center has canceled all of their fall plant sale as far as having other vendors come, having us and the Austin chapter come. They are doing a smaller scale plant sale where you can get on their website, make an appointment, and go out to their greenhouses out there, and you'll be able to shop and social distance as well. So get in touch with the Wildflower Center and go out there and get your native plants. And also you can find us at the local area farmers markets. We're not sure exactly which ones we're going to do, the dates or the times, but we are going to do one day at Sun City, and we will be doing one day at the Wolf Ranch Farmers Market. And we are looking for another market in a different area to do a plant sale. So get in touch with us if there's something special you want us to have at those markets. get in touch with Beth Irwin, or if you get it to me, I'll forward it over to Beth Irwin. And we will announce, once we have our date set, we'll announce what volunteers we're going to need. It's a still upcoming River Ranch County Park. This is a virtual field trip, and the park is still closed, but we have not been able to complete that yet, but we're still working on it. Upcoming programs, we look forward to having Jane Tillman, Native Plants for Birds. She's a big birder, and she helped develop this class. It was developed by NIPSOT in conjunction with, it was developed by NIPSOT in conjunction with the Audubon Society, but this is her own presentation of Native Plants for Birds. This is not in LCP. UT Endell, Medical Center, Native Landscaping, and then our December party, holiday party is obviously canceled. And so we are the Williamson County Chapter, Native Plant Society of Texas. If you want to find us, looks like you already have. But in addition to our website, we are on YouTube. We are posting some on Facebook, and have just begun posting on Instagram again. So whatever you like, follow us. Also, if you want to sign up for our email notifications, go to our website and we will send you announcements on our upcoming speakers, plant walks, plant sales, etc. And we do not share that list with anyone, so it's safe enough and you can easily unsubscribe. On YouTube, if you go, all our meetings we're putting on YouTube. You can look at past meetings, and this meeting will be on YouTube as well. And you can search on Facebook for NIPSOT Williamson County, and you'll find that. This evening, we are really excited to have David Lewis, some beautiful pictures of rooms. He's got a book that he has co-authored. And one lucky winner tonight. We will take a random number from the list of participants, and we'll be contacting you about sending you a book from Amazon, his book on mushrooms. So David is co-author of this book. Like I said, he has also discovered several new mushroom species, 4 of which got named for him. He's a field associate. Oh, you have signed out. Oh, signed in. Whoops. David?
Yeah, I'm here.
Okay. I think Gary just signed in. It said I signed out. I hope I'm still presenting. Okay, and we're going to switch over to David now.
Okay, are we ready?
We are.
Okay, well, first of all, thank you for inviting me to speak to your group. And I'm doing this in my son's house in College Station because I'm away from home because of the hurricane. I had probably 100-mile-per-hour winds in East Texas, and fortunately my house was spared. And to make things a little bit easier, I'm doing this from College Station where I have better internet connection. So with that, saying that, anyway, let's get on to about mushrooms. Okay, first of all, I've been studying mushrooms for about 45 years, dating back to my graduate days at Lamar University. And mushrooms and fungi have always been viewed with mystery and superstitious because they suddenly appear after rains, and then they disappear. So we really don't know too much about them without a fruiting body. And one of the things you want to do if you are interested in eating wild mushrooms is to be aware some of these picking and eating wild mushrooms can kill you. This is especially true of the genus Amanita, which we'll talk about in more detail in a few minutes. This is a poster they put out some years ago because too many people were eating the wild mushrooms in California and other places and getting their own species, and some people would actually die, and the others had to have liver transplants. So the identification of what you eat is very critical if you do this. Of course, I always get the question, is this mushroom edible? I always say every mushroom is edible once. First off, a few definitions. What is mycology? It's a branch of science for fungi or the study of fungi. And a mycologist is one having knowledge of fungi. So that's pretty straightforward. Now, the kingdom of fungi is very diverse and morphologically diverse group. As this photograph shows, there's big ones like the one on the left, Macrocybe titans. You have the small marasmoid things on the right. The bottom left, you have one of the species of chanterelle. And on the right, you have one of the artist conch, or the bracket type of fungi. One of the most sobering things about the fungi is that we know so little about them. This is a study based on the ratio of fungi to plants in England, done back in 1991. And using this ratio, if you extrapolate to a world, potentially there's more than 1.5 million species of fungi. So we just really know about 5% potentially of all the fungi known. Of course, look down at viruses, with this thing about the viruses, we only know probably about 4% of the viruses. So the small things are still relatively unknown. So basically we live on an unexplored planet still today. Now in Texas, based on some studies myself and my colleagues have done, we have about 1,400 species of just the so-called high Basidiomycetes from Texas so far. But there's a lot more we're still finding. Just a little bit about nomenclature. Basically, what we usually go by is the genus and species. That's sort of like our handle when we talk about species to other people. We just go generally with the genus Agaricus and species of bisporus. This is a common mushroom of commerce. Of course, they have their own kingdom now, the kingdom of fungi, because they differ from plants. The way they're different from plants, well, first of all, they had no chlorophyll, they had no vascular system. They reproduce by spores instead of seeds. And the so-called vegetative phase is a microscopic structure called hyphae, or a mass of mycelium. And chemically, mushrooms are composed of chitin rather than cellulose. And all the mushroom is the reproductive structure of the fungus organism. They have basically three functions in nature. One, they're saprophytic, they live off dead organic matter. Some are parasites, they attack and kill other organisms. And then the most interesting function is they're mutualists. They form symbiotic relationships with other organisms, such as lichens and mycorrhizae. Of course, a lichen is an algae in combination. The algae produces sugar, and the fungus gives the algae a structure to live in. The other one is mycorrhizal symbiosis. Y'all hearing me okay? It says my connection is okay. Never mind. Okay. Mycorrhiza is an association between higher plants and fungi. In this cross section, you can see the extensive mushroom mycelium in the soil. If you actually look a little bit closer, you'll find these swollen root tips in the soil. This is the actual physical connection between the fungus and the tree. And the tree gets nutrients and water from the fungus, and the fungus gets sugars from the tree. So it's a true symbiosis. Now, first group I want to talk about are the BASIDIOMYCOTA. These are most of the ones most people are familiar with. The BASIDIOMYCOTA have a basidium, which is a microscopic structure, and generally they have 4 spores that are shot off. A little bit about the anatomy. So you have a cap of pallius or pileus. Some have gills, others have tubes. Some have a stipe or stem, and others have an annulus or skirt. Right here's the annulus, and here's a cup-like thing called a vulva at the bottom. And of course, as a mushroom develops, it's good to get all stages of development, because when you find a mushroom, sometimes it might be too old to determine what it is, but if you find all the stages, it makes the identification a lot easier. One of the first things you want to do is make a spore print, because this helps get it in the right family. And all you need to do is silver the cap from the stem and put it down on a piece of white paper or black paper or both, and wait about 24 hours, and generally you get a spore print. And here's another example. This is a species that has a sort of yellowish spore print. But these spore prints can vary from pure white to yellow to green to black to rusty brown and so on. So obviously the color is important in their identification to families and such. Now, one of the little tricks I learned was to take mylar, pieces of mylar, and just do the spore prints on mylar, That way you can hold up the plastic against a black and white background and have a better idea about the color of the spore print. The first group we'll talk about are the agarics. These are the so-called gill mushrooms. These are the ones that have the radiating plates on the bottom where the basidia are formed and they produce the spores. And first group we're talking about is the genus Amanita, because these are some of the most feared and loved of all mushrooms. They're feared because, first off, a little bit about the vulva. The vulva is a structure that the mushroom develops in. And a lot of times they're buried in the ground. So when you collect a sample of Amanita, make sure you get the vulva out of the ground, because it's a really important identification. Anyway, as I said, some of the Amanitas are some of the most deadly mushrooms known. In this case, the destroying angel, which we do have here in great abundance. And here's just another example of some of the so-called white Amanitas. If you have a mushroom that's pure white, has the skirt, the vulva, and white spore print, leave it alone. So this is one, if you do edibility, learn it first. And those for you that are chemically inclined, this is the structure of the toxin. It's a protein, but it's very insidious because the protein is very stable when heated. So you can heat this thing, cook it, and the mushroom toxin is still there. You ingest it, and about 8 hours later, the thing's destroying your liver. So obviously, this is one you really need to know if you eat wild mushrooms. Now, on the other hand, we have one called the American Caesars. This is now called Amanita jacksonii. The true Caesars is an actual European species, and the one we have in North America is different, but it's a choice edible as long as you know what you're doing. Notice it has this kind of reddish orange cap, has striated margin, has the annulus, and the vulva is like a sac egg-like thing in the ground. Well, you say, well, that's fairly easy to tell. Well, the thing is, there's a lookalike. We have the Amanita muscaria, which is a fall mushroom. It generally has these warts on the cap. It lacks the striations, and the vulva structure is different. It has certain compounds that make you either ill or hallucinate. And I always tell people, well, they're separated in time. The muscaria is fall, the Caesars is a summer mushroom. Well, guess what? When you found both of them together, which brings up the point, although we think these things are separated in time, that's not necessarily true, especially with the warmer climate now. We're finding more summer mushrooms fruit into the fall season. Excuse me. A couple of other mushrooms. This is one called Amanita roseotincta because of the rosy salmon colors. It's found with pine trees. Another one that we have in the fall is one that's called Amanita polypyramis. This is probably one of the largest mushrooms that we have. It's sometimes the caps the foot across. Another one that's really quite interesting is one called Amanita westii. You notice the gills are kind of chocolate brown, but the spore color is white. And this mushroom was discovered back in the 30s in Florida. It was so rare it hadn't been reported for like 50, 60 years. And we found it in Texas and Mississippi in the 80s and 90s. And so now its range extends all the way from Florida to eastern Texas. Another one really interesting is Amanita thiersii. This one's actually collected here at College Station, Texas back in the 50s. It was described by Dr. Thiers as a different species, but it was found out it was not correct. So they went and named it for Dr. Thiers. And this thing is interesting because it's a non-mycorrhizal amanita. All the amanitas mostly are mycorrhizal except for this one. It actually lives off decomposing grass. And another fascinating thing about it is slowly moving. It's more or less pan-tropical, but it seems to be moving up north. We have reports of it from Illinois, Kansas, and Maryland. So it seems to be advancing north with time. Now, one that probably you see on lawns is one called Chlorophyllum melophyllidioides. This one is interesting because it has a green spore print. As you can see here, the gills have a greenish gray color to it. It has scales and annuals, but no vulva. So it's different from the amanitas. This one is one that's unfortunately is toxic. It will not kill you, but it makes you awfully sick. And I get a lot of calls from the Poison Control Center about this species. One is if you have a greenhouse or mulch, you'll see this one called Leucoprinus burbermii, a beautiful yellow mushroom. Another one that grows in the woods in the summer, Leucoprinus fragilissimus, is really real fragile. It's usually by noon it's gone because it's so fragile. Okay, next family are the Hygrophorus. They're called waxy caps because if you put the gills between your fingers and rub it, it has a waxy consistency to it. And these are white spored. They have no annulus, no vulva, and they're quite colorful. They have reds and greens and yellows and so on. A couple of examples. This is Hygroscope mississippiensis, obviously described from Mississippi, but found in Texas also. And another one is Hygrophorus chameleon, which we just found in Texas in 2007. Once again, it's described from Mississippi. And you think, well, what's the name chameleon means? Well, if you look at the left photograph, you see it has sort of a maroon, reddish maroon cap. And the one on the right is kind of faded out. But if you take the cap and lick it with your tongue, it reverts back to the original color. So hence the name chameleon. Another one that's kind of fascinating is one called Hygroscope atrosquamosa. This is a tropical species found in Martinique, Trinidad, and Puerto Rico. And we have collections of it from Galveston County, Texas now. Now, the last group, more or less white spores, are the Russulas and genus Russula and Lactarius. These are different because they have a brittle consistency. I mean, basically, you can throw them against a tree and they just sort of shatter in 1,000 pieces. The reason being, look at it under a microscope, and what's called spherocysts or round cells. So it gives it a brittle consistency. Another thing, the spores will turn blue with iodine, and the spores have spines and reticulations, which help in their identification. See a couple of examples. Well, first let's look at Lactarius, which of course it means exudes a latex. You can see the latex right here on this slide. Okay. Next are the easiest one to identify as blue Lactarius indigo. We have the summer form and the winter form. They're actually two varieties of the same species, but they're different in the time of year they fruit. Another one, winter one we have in the winter is Lactarius paradoxus. The latex is red and bruises the gills kind of a bluish green. Then we have one called Lactarius salmonius, which has these orange, beautiful orange gills. And some of the features about the Lactarius is a lot of the latex will change color or bruise the gills different color. In this case, it bruises purplish. And one of the favorites to eat is one called Lactarius bulimus. It exudes a white latex. It has a rather fishy odor to it. And you can cook it and the fish odor goes away. It's really quite an excellent edible. Okay, next is Russulas. Excuse me just a minute. Okay, this is once again another large genus which has many undescribed taxa in it. This is, they have reds and yellows and greens and purples and so on. I mean, very variable colors in the caps and so on. This is Russula flavida. One that has a really interesting odor is one called Russula mutabilis, which has an odor of benzaldehyde or almonds. One that has a sort of quilted greenish cap is one called virescens. Then we have one called Paleocrestosa, which is still undescribed. It has these rather pastel lila colors. And we're still working on getting this one published as a new species. In fact, we did publish one new to science a few years ago. This is Russula texensis. It's only known from one site, which is my property in East Texas. Another thing about the Russulas, they have these, especially the red ones, they have these pigments that if you extract them and put them under a black light, they fluoresce this really intense blue color. So it's kind of fun to play with some of these pigments and see what kind of chemistry they have. Okay, the Another group of white-spored mushrooms are the Tricholamataceae. Now, these sort of catch-all for different groups that now probably have different family names, but for right now we'll just call them all, you know, sort of a hodgepodge of different species. Of course, the oyster is one that we have that's common on oaks, willows, and so on, mostly in the wintertime. These are also, by the way, excellent edibles. And probably one of the most astounding ones we have on the Gulf Coast is one called Macrocybe pitans, and this is probably the largest mushroom in North America. These photographs were taken in Florida. We do have it in Texas, but I've never seen them this big in Texas. So Florida does have us beat on the size. But they say it's edible, but usually it's too old by the time we find it. Now we have, as I was talking about, some mushrooms are parasitic. This is one that's parasitic. This is Armillaria mellion, so-called honey mushroom, because of the kind of honey color, and they have a scrotal annulus on their stipe, and they grow, of course, on dead wood after they kill the tree. And its sister is one called Armillaria pubescens, which lacks the annulus and has more brown colors. Both of these are edible, but you have to really cook them well because they have really a tough consistency to them. Now, pubescens is real common in the fall when you have, after you have a fall rain. But the thing is, once again, be careful. There's one, so-called deadly gallerina. Okay, it grows on wood. Got the skirt right here. You can barely see it, but anyway, it has a rusty brown spore print, so that's one easy way to tell it apart. But the thing is, it does have the aminotoxins, so I should be really careful about eating brown mushrooms. A couple of other harassments, ferruginous, which decomposes leaf litter. We have the little mycenas. This is one grown on the base of a pine tree, which has a odor of watermelon, which is really quite pleasant. And then we have one, Macrocybe texensis, which we discovered over in the Big Thicket a number of years ago. And we named it for Texas because it's so far only known from Texas and Louisiana. Okay, up to now I've talked about white-spored mushrooms. Now let's look at some of the other species that have different color spore prints. This is Genus Cortinarius.
David, we've got a couple of questions about spore prints before you go too much further.
Oh yeah, sure. Okay. Go ahead, Gary.
So I got a question from Sue, who's asking, can you do spore prints? Do all mushrooms have spore prints?
They should, yes. Sometimes they won't. They're either sterile or they're too old to put out spores. But yeah, get a free body that's it's fairly good shape, and chances are you get a spore print on it.
Okay. And then Martin asks, for the spore prints, when you're trying to differentiate species, is there anything that you're looking for besides color?
Oh, you mean? Oh yeah, of course. I mean, that color is just one of the things that helps get it to a family or a genus.
So it's when you're doing a spore print, you're really just checking for color to kind of help guide you.
Yeah, to help guide, get in the right family or the right genus. Correct. Yes.
Okay. All right. Thank you.
Oh yeah. Okay. Okay. Now, anyway, Cortinarius is one of the largest genera of fungi in the world. Very difficult to get down to species. We probably have over 100 species on the Gulf Coast. The main thing is they have this little web-like thing on the stump called a cortina. And the cortina has a habit of catching the spore. So you have a ready-made spore print on the cortina if you look real close. So that's kind of handy. Usually a cart will have the cortina with a spore, so you have a Cortinarius. One that's really fascinating is one called Cortinarius marylandensis because it has these red pigments. And you take it and you can extract the pigments and then treat it with different metal salts like alum or whatever. And you get these different colors. All these colors come from that one mushroom based on using different types of metal salts. So there's a whole subculture in mycology that they dye with mushrooms. It's DYE. And speaking about dye, this is an interesting one. This has a rather interesting ecological niche. It grows around mammalian corpses. And we call it the corpse finder. I don't use common names often, but this was too much. This was just too much to pass up. But anyway, it uses ammonia. It needs nitrogen from the decaying protein. That's how it gets it. So if you have well, as an example, after one of the hurricanes, we buried some food in the yard. A year later, we had these hebelomas coming up where we buried the food. So apparently the spores are in the ground, quite readily. A couple others. This is the genus mutabilis, which has a rusty brown print. Some of them grow on pine cones and pine detritus, and some grow on hardwoods and so on. Then we have the so-called little brown mushrooms. These are ones that are really small, and they're really sometimes tough to identify, but this is one we know. It's called Galleron grows in sphagnum bogs. And then we have this other one grows in bogs called Galleron. And these are, once again, some of these Gallerinas do have the toxins, aminotoxins, so they're too small to fool with for edibility, but they're best left alone. Another one, Genus Inocybe, they're called fiber heads because if you look real close at the cap, you see they have these fibers that break out from the center. This is a large genus. They're ectomycorrhizal with probably hardwoods, basically. And they're superficially, they look alike, but once you look at the microscopy, they have different types of spores and different type of microscopic structures. So microscopically, they're quite fascinating. Okay, and finally we have the chocolate brown spore, the genus Agaricus. This is one we're all familiar with, bought in the grocery store. And we do have wild ones here. And most of the wild ones, if they don't smell medicinal or phenolic or don't bruise yellow, they're probably okay to eat. And this is another species that we found on lawns. And then I said about some of the wild ones, some of them are a lot more tasty than the one you buy in the store too. Okay, this brings us to an interesting subject, the discovery of mushrooms that cause strange visions. I don't know how many of you are aware of the ones that people used to go out in cow pastures and collect and make teas out of. This is mostly back in the 70s and 80s. Well, what brought all this about was this article in Life magazine in 1957. It was written by Gordon Watson, who was a banker. He was a banker, but his wife was Russian and she loved mushrooms. And it led to him becoming interested in mushrooms, and he became actually the first ethnomycologist. Well, he and some other gentlemen went down to Oaxaca, Mexico, because they heard about the religious rituals that these people were using in Mexico. And he more or less rediscovered this mushroom cult down in Mexico. And this is a couple of the watercolors in the book, the Life magazine. I mean, they're really quite exquisite drawings. And of course, the one we have here is, if you have a cow pasture, chances are you'll see this in the cow pasture. And it will turn blue, usually on the stipe. And it has sort of a purplish brown spore print and kind of a golden colored cap. And we have this one, which is found in Alabama, Louisiana, Texas, and Mexico. It's quite rare. I've only collected it 2 times in 45 years, but you can see the blue in reaction right up here on the upper part of the stipe. One group that has what's called a pinkish spore print is the genus Untitled. These are terrestrial mushrooms. You can actually see the spore print right here on the cap of the one below this one. These are interesting because the spores are angular under a microscope, so they have some interesting spore morphology. One we just found in Texas a few years ago was this Untitled incanatum, which has this kind of yellow color. It bruises kind of deep green, blue-green, and it has an odor of mouse urine. Now, the next group are mushrooms with tubes. These are the boletes. This is a highly esteemed group. Most are edible in Texas. Once again, you see the tubes, and some of them were bruised blue. Actually, the tube structure in this photograph. Now, this balloon reaction is different than the magic mushroom, so it's 2 different types of chemistry. And a lot of these will have reticulated stipes. That sometimes helps in their identification. We probably have, I don't know, maybe 150 species of boletes in Texas, probably. What is really nice to find is one called Pseudopinophyllus. You see the reticulated stipe, kind of this reddish brown cap, and this is probably one of the best edibles that we have. Generally, it grows with pine trees, as the name implies. The other one grows with live oak, and you should have this if you have live oaks in your area. Look for this one. It's right this time of year after the rains, Lourdellis. Described from Florida, but you can see the reticulated stipe right here. Has sort of a brownish cap, and the stipe the bottom was bruised, kind of a reddish color with age. But sometimes you find this in great numbers. You can dry them out and save them for making soups and such as that. It's probably one of the best edibles that we have, and it's pretty widespread with live oaks. One that we named a couple of years ago was Boletus rudii. Our colleague Bill Rudy discovered it in West Virginia a number of years ago, and we kicked it around a few years, and finally we published it, named it for him because he found it. But it's found from West Virginia to Arkansas down to Texas, and probably all along the Gulf Coast too, I suspect. One that's rather fascinating was one, the genus Hygrophorus. This one has both a tube-like and a gill structure to it. So this is our missing link in mycology right here, between the boletes and the gill mushrooms, so to speak. Okay, now the chanterelles. These are some of the most highly esteemed mushrooms that we have. You have them out there. I have collections from around Austin and such. Trouble is I cannot put names on them because some of the ones out there are probably undescribed. But a couple that we do know, this is Cantharellus Lactarius, which has this smooth underside. It doesn't have true gills. It has ridges and such. And this taken on my property in East Texas. I mean, sometimes it's just everywhere. The young lady is a Swiss mycologist. Her and her research friend Bart Buick are the world's experts on this genus, and they've spent a lot of time with us in Texas. And a lot of these new species were named by Bart Buick from Paris, France. This one he named. This is Cantharellus altipes. That grows with pine trees. And then we have this is a no name, Cantharellus cinnabarinus because of the cinnabar color. But my colleague was doing DNA, and he found this very similar look-alike called Cantharellus texensis. And the main difference is the DNA is different, and the spores are a little bit different. Other than that, they look just alike. So once again, it shows you some of the subtle things that you have to look at when you do mushrooms. By the way, all these chanterelles are edible, by the way. One that we found a couple of years ago, and actually we found it in Louisiana and also Jasper County, Texas. But the collection was mostly, the best collection was from Louisiana, where we named it from. So we called it Ludovicianus, which means of Louisiana. But we do have collections in Texas also. Now, we'll move on to some other groups. These are the so-called shelf fungi, bracket fungi, whatever, polypores. Now, they grow on wood. They're tough. They have a tough consistency. They're not mycorrhizal. They're mostly saprophytic or parasitic. A couple of examples. This is the so-called true turkey tail because it has these concentric rings on the upper part. If you look closely, you see they have pores. Okay. If you look, there's a look-alike called the false turkey tail, Genus. If you look at the bottom, it has a really smooth surface on the bottom. Then we have the coral fungi, as the name implies, it looks like a coral. Then we have one that looks like a cauliflower, the genus Spiroceus. It looks like bacon, but this is edible, edible by the way. Then we have the tooth fungi, the genus Hericium, which has these long tooth-like structures. And they generally grow on oak trees, sometimes 20 or 30 feet off the ground. And once again, this is an easy one to identify. It's edible. We usually cook it, cut it up in pieces and roast it in the oven. It has a taste like lobster. Then we have the so-called jelly fungi. These will dry up between rains, and when they get rain, they rehydrate and put out more spores. This one's actually edible, by the way. And then we have the puffballs. Now, the puffballs differ because the spores are born internally, unlike the other mushrooms, which they're born externally on the gills or tubes or teeth. So it takes a mechanical strike, stomp them, or a raindrop or an animal stepping on them to help push the spores out of the fungus fruiting body. A couple of examples. This is the genus Calvatia. You can cut it in half. If it's pure white inside, it's good edible. Once this turns just a little bit of yellow or brownish, it's too far gone. But once again, there's a little caveat. Make sure you cut it in half. You don't see the button stage of an Amanita mushroom, because this is the button stage of an Amanita mushroom. So it looks like a puffball when you first see it. Okay, another one you be aware of is the so-called earth balls. These called in genus Scleroderma, which means hard skin. And generally they have this purple interior, which is a dead giveaway. These called gastrointestinal upsets. Another interesting one is one of the so-called stalk puffballs, genus Calistoma. And we have the earth stars. As the name implies, it looks like a star. And then we have the stink orange. If you have mulch in the wintertime, chances are you've seen this thing. But well, first off, usually you smell it before you see it. But of course, they're important in decomposing mulch and such as that. Okay, now the last major group are the Ascomycota. Now this is the differs from the basidials by having an ascus, which is a sac-like structure, and generally have 8 spores, which are shot out. And this is a large group of fungi. I mean, think about penicillin. Penicillin is in this group. Of course, our yeast are Ascomycetes. Of course, this is probably one of the most important, economically important organisms on the planet when you think about bread making, wine making, fermentation, and so on. Morels, now you're in the Morel belt where you live around Georgetown and Austin and Waco. I mean, I don't see them in East Texas, but you have the right time of year you can find morels generally March to 1st of April in your area with adequate rainfall naturally. A couple of interesting ecological niches. This is Hygrophorus, which grows parasitically on gilled mushrooms. You can actually see them develop from the mushroom right here. It's white, you see the gills, and it slowly takes over the gill structure. And this one is considered edible, but you're never quite sure if it's about the host. I've eaten it, and I have had no problems, but be aware. Another fascinating one we found a year or two ago is this Spherostobelia. It was found grown on the bottom of it's actually a hyperparasite. It was grown on the bottom of a polypore. We sent it to specialists, and they said it was new to science, so we just published it last year. And epithet toxico means they found out it was toxic to both bacteria and viruses. So there's always some potential medicinal benefits to these things we just aren't aware of yet. Another interesting niche. This is a cordyceps, which grows parasitically on insect. This is some kind of fly. Here's the fruiting body right here. And we have others that actually parasitize truffles. This is a false truffle. This is the parasite on the truffle. A couple of interesting habitats. These are the genus Zolaria. One grows on magnolia cones, the other one grows on sweet cone balls, and it's 2 different species. And one that you have in the area, this is Corioastus giaster, the Texas devil cigar. I had reports, I think, Bastrop, and all the way up to Dallas. So it's almost endemic to Texas, and of all places, Japan. It's only known from 2 places, Texas, Central Texas, and Japan. So it's sort of a very disjunct population. And of course, we do have truffles. This is the truffle which grows with pecan trees. And they actually harvest this in Georgia under the pecan trees. Now, I'm sure we have them in Texas. I just don't really look for them that hard. But if you have a pecan orchard, take a look and see if you can find it. And as I mentioned in the first, there's, we still have a lot to learn about mushrooms and the ones that are out there. This is a species of truffle I collected in my front yard a number of years ago. I sent it to a specialist, and he says, well, it's new to science, but we need more material to properly describe it. Now, unfortunately, I haven't seen it since, but it just shows you some of the problems about doing taxonomy. And I think that's all.
All right. Thanks so much, David. There's a
Oh, here's one more. Here's some useful field. I'm sorry.
perfect.
Useful field guides.
And unfortunately, I missed the very beginning. Did Randy mention that somebody was going to come away with a copy
Oh,
of
yeah.
the book?
Yeah.
Okay.
Yes. Well, good.
Okay.
Oh, by the way, let me mention one more book. This one just came out, Amanitas North America by Brett Barnyard and Jay Justice. So actually, if you want to study Amanitas, this thing is great. It has a tremendous amount of information. Just came out a couple months ago. So let me mention that too. And I see, I think I have one. Oh yeah, a couple websites that are quite useful. And I guess you'll have a copy of this, won't you, so people can look at it.
Right. We'll post a copy of the recording to YouTube, and then I'll try to remember to pull out some of these links and put them in the description on YouTube.
Okay, great. Thank you. Yeah,
And
I think
then,
it's, yeah, okay. Okay, what do I do now?
no worries. You can stop sharing your screen, or and then we
Okay.
can go through some of the questions.
Oh, there we go. Okay.
Okay, perfect. Now that you're not sharing your screen, it's very dark on your side.
Well, I know I have all my lights off, so yeah, I have only sunlight coming in, what little sunlight there is.
Oh, no worries, no worries. Okay, so I'll try to summarize a couple of the questions here.
Okay.
Let's see. One from Sue says, "Are you, when you're identifying the mushrooms, how successful are you at identifying them without removing them?"
Well, if it's one that's pretty obvious that I know, I'll leave it. But if it's something unusual or I don't know, I'll collect it. I mean, a lot of times I'll just make notes like, gosh, some of the chanterelles, there's so many, like the Lactarius. Of course, it's edible, but sometimes there's so many, you just leave them in place. And you actually, by collecting mushroom, you usually don't see the mycelium is in the ground, so you're not really hurting the organism. You're just taking the fruiting body off.
Okay. So it's just the fruiting body that's getting removed when you're doing that.
Right, yes. And some people will take scissors and just cut it so they leave the base of the stipe in the ground so they won't disturb the soil.
Okay, awesome. And then I've got a question for Martin that I'm probably going to manipulate just a little bit.
Okay.
I think it's about for the species of fungi. What would you say might be the percentage of those that produce recognizable mushrooms when they fruit?
Oh, goodness. That's a good question. I don't think I can answer that. I mean, there's a lot of, let me give you an example. We did I had some colleagues do a survey on my property in East Texas. They took soil samples and did what's called blast DNA. They look at and potentially, he says, I have 2,500 species of fungi in my front yard based on DNA. So the thing is, until I get a fruiting body, I cannot put a name on it or even start to classify it. Does that sort of answer the question?
I think so. I think what that tells me is, as a novice person wandering around, I need to be very, very careful, especially if I'm thinking about ingesting this mushroom.
Oh, right, yeah, because I would suspect, geez, I really can't put, I'd say at least 10 to 20% of the mushrooms in Texas are still unknown. I mean, that's just a wild guess, but based on, previous studies and new species, in the last 20, 30 years, between myself and my colleagues, we've described 30 new species from East Texas. So that just gives you an idea of what's out there.
No, that's certainly interesting. Do you think is it lack of study or new interest?
Yes, yeah. Well, we just don't have too many mycologists. I mean, the universities aren't funding taxonomy like they used to, and most of the work now is done by amateurs.
Okay.
And the amateurs are quite good too, by the way.
We'd like to think so, at least for the Native Plant Society.
Oh yeah, definitely. Well, definitely, yes.
Um, do you use any sort of online tools for folks? Would they have luck if they were to take pretty decent photos of a mushroom that they encounter and post to iNaturalist? Do folks watch that? Some of the
Yeah,
mycologists?
yeah, I get usually, myself, I don't really, I'm so inundated with my own mature right now. I do IDs, you know, people email me. We have there's mushroom, what's it, Mushroom Expert. There's a couple of websites besides our naturalists, and generally there's enough people out there, you know, we can sometimes figure it out. I'm trying to think of this other website, Mushroom Observer, I'm sorry. But that's out there, and I guess you can post on that, and people will, you know, comment on what you have.
Okay, Mushroom Observer.
Mm-hmm.
All right, it sounds like a journal. All right, and let's see. Oh, the other question I had is one that I raised while we were doing our preliminary. You know, we kind of talk about native plants specifically and the dangers of bringing, you know, plants that are invasive or not natural to a particular area. And I believe you mentioned that that's going to be true for fungus as well. There's
Oh,
things
certainly.
here in the U.S. that didn't belong.
Yeah. Prime example is the death cap, Amanita phalloides. It's a European species, and it was brought over on probably root tips of some introduced plant in the Northeast, and it's spread, I think, Delaware, New Jersey area. I'm not sure how extensive it has, but it has spread along the Atlantic coast up in the Northeast. So yeah, you can have invasive mushrooms.
Okay. All right. We've got time for just maybe a couple of more questions. I've got one here from Kathy McCormick. Is dodder a mushroom or a fungus?
What now?
Dodder, D-O-D-D-E-R.
Oh, that's a plant. It's a parasitic plant.
Parasitic plant. Okay. Thank you. And then I'm going to really butcher this the scientific name. Let's see. What is the status of Meristoma coliforme, the salt shaker earth star in Texas?
Yeah, I don't know if I have any collections or not, to tell you the truth. I'd have to look at my database and see. Myself, I've never collected it in Texas.
Okay.
That's not to say someone did. There was a Dr. Long back in the 1900s that collected puffballs and stinkhorns around Austin and Denton. And I have all his literature. I can look it up, but I don't have the access to it just this minute.
Okay, but, no problem.
Yeah,
Elizabeth goes on to say that she knows that in Europe it's considered rare and threatened, and was just kind of curious if that was the same case here in Texas or in the U.S.
Oh, well, since we have no data, who knows? I mean, it's a trouble. A lot of this is we don't really know because we don't have the information.
Okay. All right. Well, I think that's probably got our time. I think Randy had to drop, so she's my timekeeper usually. So thanks, David, so much for all of this. And then
appreciate y'all listening, and thank y'all for buying the book.
All right. Well, thank you for doing this. It's definitely an interesting topic.
Hopefully next year we can all get together physically.
Right. No, we're looking forward to that as well.
Yeah.
All right. If we have any additional questions that kind of come through, I might send those over to you and let you respond to them. And then if I see anything that comes through on YouTube, we might toss some of those over to you as well.
Oh, okay. No problem. I'm glad to look at them.
All right. Well, thank you so much, sir. You have a good night.
Okay. Goodbye, everybody.
All right. Bye-bye. Thanks, everyone.